Now, let's look at the mechanism of action of pyruvate dehydrogenase complex. Initially, we say it's just the removal of CO2 and subsequent addition of coenzyme A to pyruvate that gives us acetyl-CoA, but it is not that simple. We see the products CO2, NADH plus hydrogen. How does this actually happen? In this complex that contains 3 enzymes in one. Let's see that. The first enzyme of this pathway, the pyruvate dehydrogenase, catalyzes the removal of CO2 from pyruvate. So we have pyruvate, we have pyruvate right here. This enzyme, pyruvate, catalyzes the removal of CO2. So it removes this from pyruvate. And then we have this ethyl acetyl group remaining. So CO2 leaves, and TPP is a coenzyme of this enzyme. So CO2 leaves, and the remainder is attached to TPP. And it becomes acetyl TPP. That's the first step. In the next step, because these reaction steps are 5 in number, step 2, the mechanism occurs in 5 steps. Step 1, pyruvate dehydrogenase removes CO2 from pyruvate, and the remainder is attached to TPP, its coenzyme, converting TPP to acetyl TPP. Now, the next step is catalyzed by the next enzyme, dihydrolipoyl transacetylate. What happens there? The TPP transfers the acetate group from pyruvate to lipoate, or what we call lipoamide, or what we call lipoid. So lipoid comes in, collects the acetate group from TPP, and TPP returns to its original form to continue the next phase of another pyruvate coming in to be converted to acetyl-CoA. So it becomes acetyl-lipoamide, no longer ordinary lipoamide. Why TPP becomes free again. Step 2. At step 3, this acetyl-lipoamide is also transferred to coenzyme A, catalyzed by the same enzyme, dihydrolipoyl. So whatever it has received from TPP, it now transfers to acetyl-CoA. So we now have CoA, S, and then we have our CH3. So all of this comes in here. This hydrogen is still pending, and the hydrogen from here is still pending. So we have all of that come in here. See, this acetyl is transferred to CoA to become acetyl-CoA. And then the hydrogen from CoA and the hydrogen from the acetyl group from pyruvate are transferred to lipoid. So lipoid keeps the hydrogen group, one from acetyl-CoA from here and one from the acetyl remains, while the other part of it, the acetate, is transferred to this to give acetyl-CoA. So those 2 hydrogen left behind converts this to what? Dihydrolipoyl as step 3. Step 4 of the reaction, this 2 hydrogen here is transferred to FAD. FAD comes in, collects the 2 hydrogen from dihydrolipoyl and becomes FADH2. This step 4 is catalyzed by the last enzyme of the complex, dihydrolipoyl dehydrogenase. So dihydrolipoyl releases the 2 hydrogen from this step to FAD, to becomes FADH, and then returns to its original state, lipoamide or lipoid, to continue with another round of this reaction. So we have FADH2 as the product as step 4. This same enzyme that catalyzes step 4 also catalyzes step 5, which is what? The FADH2. FAD transfers the 2 hydrogen atoms from it to NAD to give us NADH plus H. So FAD receives the hydrogen as step 4 and then transfers it to NAD as step 5 to give us a final product, NADH plus hydrogen. So iron. So we see now that 5 of these enzymatic reactions that are catalyzed by pyruvate dehydrogenase complex is actually handled by 3 enzymes that make up the dehydrogenase complex. The first enzyme catalyzes pyruvate dehydrogenase, catalyzes this reaction. Step 1. Step 2 and 3 are catalyzed by dihydrolipoyl transacetylase, and step 4 and 5 are catalyzed by dihydrolipoyl dehydrogenase. So you can see how unique the pyruvate dehydrogenase complex acts in converting pyruvate to acetyl-CoA.